Hello students, welcome to EPG Pathshala. I am Rashmi Sinha from Indira Gandhi National Open University. Today we are going to talk on the module Causes of Human Variation from the paper Physical or Biological Anthropology. What are we going to learn in this module? It will help us to provide an introductory overview of human variation and its causes. Second, it will provide us an historical overview of the causes of human variation. 3. It will familiarize us with the adaptive significance of these variation. 4th, to discuss the various forms of variation and genetic factors that shape the human variation, and finally to provide contemporary interpretation of human variation. What is human variation and what are its causes? In this module, we shall find out some of the ways and means by which humans differ from another, both within the population and between the population. No two human beings are genetically identical, even the monozygotic twins who develop from the one zygote. Therefore, we can imagine the extent and scope for the difference of variation in and within a population. All humans are different from each other, and much of this difference has a genetic basis. Difference in phenotype caused by differences in the genotype. Some of these differences are easily observable, and we know from our own experience that they run in families, hair, eye, skin color, stature, and some morphological feature. Humans are one of the most morphological variable species of animals, and part of the reason for this variation exists is because members of our species occupy many diverse habitats. That is, they stay in such different habitats, right from cold to hot to high altitude. So many different habitats does a human species is found in. Yet the exact genetic basis of this variation must be quite small. Biochemical evidence shows that human and chimpanzees differ genetically by between 1 to 2%, and that this amount of difference was accumulated over a period of approximately 7 million years. The issue of human variation is very complicated, and the biological and cultural factors that have contributed to that variation and that continue to influence it are manifold. Historical views of human variation. Historically, when different groups of people came in contact with one another, they offered explanations for the phenotypic variation they observed. It was because some phenotypic characters such as skin color was obviously noticeable. Skin color was one of the more frequently explained traits, and most systems of racial classification were based on it. Way back in 1350 BC, the ancient Egyptians had classified human on the basis of skin color: red for Egyptian, yellow for the people to the east, while those to the north, and black for Africans from the south. In the 16th century, after the discovery of New World, Europe embarked on a period of intense exploration and colonization in both the New and the Old World. Resulting from that, contact was an increased awareness of human diversity. The first scientific attempt to categorize the newly discovered variation among humans was Linnaeus' taxonomic classification, which placed human into 4 separate groupings. Linnaeus assigned behavioral and intellectual qualities to each group, with the least complementary descriptions going to the black Africans. Blumenbach, a German anatomist, classified human into 5 categories or races, although Blumenbach's categories came to be described simply as white, yellow, red, black and brown. He also used criteria other than the skin color. Moreover, he emphasized that divisions based on skin color were arbitrary and that many traits, including skin color, weren't discrete phenomena. In 1842, Anders Ritz, a Swedish anatomist, developed a cephalic index as a method of describing the shape of the head. The cephalic index derived by dividing maximum head breadth by maximum length and multiplying by 100 gives the ratio of head breadth to length. Individuals with the index of 75 had long, narrow heads and were termed as dolichocephalic. Brachycephalic individuals with broad heads had an index of over 80, and those whose indices were between 75 and 80 were mesocephalic. In discussions on human variation, people have traditionally clumped together various attributes such as the skin color, shape of the face, shape of the nose, hair color, hair form, that is, it could be curly or straight, and eye color. People possessing particular combinations of these and other traits have been placed together into categories associated with specific geographical localities. Such categories are called races. While race is usually a term with biological connotation, it is also one with enormous social significance. Moreover, there is still a widespread perception that there is an association between certain physical traits, skin color in particular, and numerous cultural attributes such as language, occupational preferences, or even mortality. Therefore, in many cultural contexts, a person's social identity is strongly influenced by the manner in which he or she expresses those physical traits traditionally used to define racial groups. Objections to racial taxonomies have also been raised because classification schemes are typological in nature, meaning that categories are discrete and based on stereotype or ideas that comprise a specific set of traits. Thus, in general, typologies are inherently misleading because there are always many individuals in any grouping who do not conform to all aspects of a particular type. Prior to the World War II, most studies of human variation focused on visible phenotypic variation between large, geographically defined populations, and these studies were largely descriptive. Since World War II, the emphasis had shifted to the examination of difference in allele frequencies within and between population as well as adaptive significance of phenotypic and genotypic variation. This shift in focus occurred partly as a result of modern synthesis in biology and because of advances in genetics. Racism is based on the false belief that intellect and various cultural attributes are inherited along with physical characteristics. Such beliefs also commonly rest on the assumption that one's own group is superior to the other. On the matter of human origin, can the religious doctrine of original sin be reconciled with what contemporary biology says? The doctrine requires a descent from a single original ancestor, whereas contemporary biologists hold that the genetic evidence indicates that modern humans descended from a population of at least several thousand individuals. Monogenism and Polygenism. The dominant debate concerning human origin and variation before and around Darwin's time was between supporters of unitary, that is the monogenic, and of separate, that is the polygenic origin of human species. Debate centered on whether the differences seen in modern populations were of such degree that different human groups should be considered separate species. Monogenists believed that all human races were descended from one pair, that is the Adam and Eve. But then they differed from one another because they occupied different habitats. It emphasized the similarities among populations and pointed out to the criteria of interfertility between individuals of different groups. This concept was an attempt to explain phenotypic variation between populations but did not imply evolutionary change. The polygenist view, on the other hand, argued that human races or all populations were not all descended from Adam and Eve. Instead, there had been several original human pairs, each giving rise to different groups. Also, polygenists saw such a wide gap in the physical, mental, and moral attributes between themselves and other people that they were sure that outsiders belonged to a different species. Thus, human races were considered to be separate species. Adaptive significance of human variation. Human variation is viewed by physical anthropologists as a result of evolutionary factors such as mutation, genetic drift, gene flow, and adaptation to environmental conditions both past and present. We must also recognize that cultural adaptation have also played an important role in the evolution of human. Adaptation is defined as a functional response to environmental conditions in populations and individuals. In a narrower sense, adaptation refers to long-term evolutionary, that is, genetic changes that characterize all individuals within a population or species. Biological anthropologists today recognize three levels of adaptation, that is, the genetic, short-term, and developmental. The sickle cell example is an example of genetic adaptation. The way our bodies adapt to the cold illustrates a short-term physiological response. There is evidence to show that the capacities of adult physiology to adapt to new environmental stresses are narrower than the original genetic potential of a child. In any case, our bodies remain biologically equipped to adjust automatically to a range of temperatures. This physiological flexibility, which allows us to respond to environmental stresses such as temperature changes, is called the plasticity. Shivering is an example of acclimatization, a change in a way the body functions in response to physical stress. Some acclimatization responses disappear when the physical stress disappears. We stop shivering when we put on more clothes or air around us warms up. Other forms of acclimatization are longer lasting than the shivering response. Some environment in which human population lives, such as the highlands of the Andes Mountains in South America or the highlands of Himalayas in Asia, they are characterized by hypoxia, that is, less oxygen is available to breathe than at the lower altitude. Studies have shown that people who grow up in high altitude adapt to lower oxygen levels by developing greater chest dimensions and lung capacities than to people living at low altitude. These changes, a form of developmental acclimatization, are apparently not due to genetic factors. Instead, they occur when the body is challenged by low level of oxygen in environment. Studies have shown that individuals who were not born in such an environment increased in chest dimension and lung capacity the longer they lived in such environments and younger they were when moved there. Forms of variation. Like any other living organism, human beings show variation. If we consider almost any characteristic, we will find differences between various people in the population. There are two forms of variation: continuous and discontinuous variation. Characteristics showing continuous variation vary in a general way, with the broad range and many intermediate values between the extremes. Height is an example of a continuously variable characteristic as long as we consider a consistent sample. For example, a large number of people of a particular age and sex, it is usually difficult to give a straightforward explanation of a genetic basis for these continuously variable characteristics because they result from a combination of genetic factors as well as the environmental factors. Characteristics showing discontinuous variation fall into few very distinct classes. The ability to roll the tongue and blood groups are examples of discontinuous variation. These characteristics can be explained much more easily by simple rules of genetics and are less likely to be affected by other factors. Some of the characteristics possessed by an individual in a population can be said to be inherited, that is, derived from the previous generation. These characteristics are passed on in a fairly predictable way as a result of sexual reproduction. Sexual reproduction also introduces an element of randomness so that variation is brought about in a population. These two almost contradictory factors, dependable inheritance of characteristics from parents and variation within the population, are essential to an understanding of the process of evolution. Causes of variation or otherwise genetic factors that shape the microevolutionary processes which bring about variation at a population level and eventually are responsible for macro level evolution. Causes of human variation. Now, what are the different causes for human variation? Number one, mutation. Mutation is defined as a change in DNA, that is the deoxyribonucleic acid, that can occur either as a single base substitution or at a larger chromosomal level. From an evolutionary perspective, mutation is only way totally new variation can be produced. Effects on any gene should be minor. However, since mutation rates for any given locus are quite low, that is, they are estimated at about 1 per 10,000 gametes per generation. In fact, because mutation occurs so infrequently at any particular locus, it would rarely have any significance on allele frequency. Certain mutations occur every generation, but unless we sample a huge number of subjects, we are unlikely to detect any noticeable effect. In this figure, you can see the types of mutation, the gene mutation and the chromosomal mutation. What happens in the gene mutation? There is a replacement of one nucleotide by another, or there is an addition or deletion of nucleotide. Whereas in chromosomal mutation, there is a change in number of chromosomes, or there is a change in the structure of the chromosome. However, because we each have many loci, we all possess numerous mutations that have accumulated over recent generations. Most of these are not expressed in phenotype but are hidden in recessive alleles. Some examples of such a recessive mutation are the alleles of phenylketonuria, cystic fibrosis, albinism, sickle cell anemia, and thalassemia. About 1 in 12,000 babies born in the United States and 1 in 18,300 babies born in India carry this phenylketonuria allele in homozygous form and thus are affected phenotypically. Without early detection and treatment, this condition leads to severe mental retardation. Several dominant alleles that produce phenotypic effects are well known. Some examples of such a condition are a type of dwarfism, which we called as achondroplasia, brachydactyly, that in this case there are shortened fingers and toes. The Huntington disease, in this there is a progressive degeneration of nervous system which is accompanied by dementia and seizures. Gene flow. Gene flow is the movement of alleles between population. The term migration is frequently used synonymously with the gene flow. However, migration strictly defined means movement of people, whereas gene flow refers to exchange of genes, which can occur only if the migrants inbreed. This figure shows you the gene flow, how from population A and B there is a gene flow. Biological relationship between interbreeding human groups are best understood in terms of gene flow between superficially distinct populations whose gene pool overlap considerably. For example, we know that individuals from European and African population have interbred considerably since Europeans brought the first Africans to the New World as slaves. Similar processes have mixed the genes of these and other in migrating populations with the genes of indigenous American populations. These are the examples of gene flow among population of a single species that had experienced relative isolation in the past, but that continues to exchange enough genes, often enough with neighboring populations, to preserve an overall species identity. However, we must keep in mind that it would be misconception to conclude that human gene flow can occur only through a large-scale movement of groups. In fact, significant alterations in allele frequency can come about through long-term pattern of mate selection. If exchanges of mates are consistently in one direction over a long period of time, allele frequency will ultimately be altered. Due to the demographic, social and economic pressure, an individual may choose a mate from outside the immediate vicinity. Another way is the genetic drift. Genetic drift. Genetic drift is a chance factor in evolution and is tied directly to population size. The term drift is used because as a completely random process, the allele frequencies can change in any direction. A particular kind of drift seen in modern population is called founder effect or bottleneck effect. Founder effect operates when an unusually small number of individuals contribute gene to the next generation, making for genetic bottleneck. This phenomena can occur when a small migrant band of founders colonizes a new and separate area away from the parent group. Small founding population may also be left as remnants when famine, plague, or war ravages a normally larger group. In another sense, we can say that each generation is founder of all the succeeding generation in a population. This figure shows the genetic drift, how the bottleneck effect is shown here. One of the numerous examples of genetic drift that have occurred in human history began early in 19th century when British soldiers occupied the island of Tristan da Cunha in the Atlantic Ocean. Eventually, the soldiers withdrew, leaving only a single married couple who were later joined by few other settlers. Throughout the 19th century, the population of Tristan da Cunha never grew much beyond 100 individuals. Over the 20th century, the population has grown to as many as 270 people, all of whom owe an enormous proportion of their genes to very few individuals. It was calculated that more than 29% of those living on the island in 1961 had gene contributed by two of the original founding population. Another cause for human variation is the natural selection. Natural selection is a mechanism of evolutionary change first articulated by Charles Darwin. It refers to a genetic change or changes in frequencies of certain traits in a population due to differential reproductive success between individuals. This figure shows the natural selection. The best documented case in case of human concerns the sickle cell allele, which is result of single amino acid substitution in the hemoglobin molecule. In many human populations, only one allele, that is the hemoglobin A, is present. In other populations, however, mutant forms of the hemoglobin A may also be present. One such mutant allele, known as HBS, alters the structure of red blood cells, distorting them into a characteristic sickle shape and reducing their ability to carry oxygen. When individuals inherit the HBS, the allele, from both parents, they develop sickle cell anemia. About 85% of those with HBS genotype, that is in a homozygous state, do not survive to adulthood and hence do not reproduce. Because the HBS allele seems to be harmful, we would expect it to be eliminated through natural selection. But in some population of the world, it has a frequency of up to 20% in the gene pool. Why should that be? Geneticists might have concluded that this high frequency was a result of genetic drift if it were not for the fact that the areas with high frequency was a result of a genetic drift. Why should that be? Geneticists might have concluded that this high frequency was a result of genetic drift if it were not for the fact that the areas with high frequency of HBS are also areas where mosquito-borne malaria parasite is common. There is in fact a connection. People exposed to malaria have a better chance of resisting the parasite if the hemoglobin genotype is HbA-HbS rather than the normal HbA-HbA. This is an example of what geneticists call as balanced polymorphism, in which the heterozygous genotype is fitter than either of the homozygous genotypes. The HbA and HbS allele are co-dominant, with the result of a single HbS allele change the structure of the red blood cells enough to inhibit malarial parasite but not enough parasites to cause sickle cell anemia. Now this table shows the effects of four evolutionary processes: the mutation, gene flow, genetic drift and natural selection within and between the population. Contemporary interpretation of human variation. Physical anthropologists and other biologists who study modern human variation have largely abandoned the traditional perspective of describing superficial phenotypic characteristic in favor of measuring actual genetic characteristic. Those traits that differ in expression among various populations and between individuals are more important in contemporary studies of human variation. Such characteristics with different phenotypic expressions are called polymorphism. Human blood group is one genetic trait with different alleles. Each person has one of the four blood types, that is the A, B, AB or O, and these four types comprise the ABO blood group system, which was first discovered in 1900. Because it has more than two variants, a genetic trait such as this one is called polymorphism. That is, poly means many, morph means form. Each person has one A, B or O allele on one chromosome of the homologous pair and another ABO allele on the other chromosome of that pair. The combination determines the person's blood type. Some of the most exciting contemporary DNA research has revealed a whole new array of genetic markers. Showing a tremendous amount of variation within and between population, these single nucleotide polymorphisms are known from well over 10 lakh sites on the human genome. Closer examination of human genome has also revealed that DNA segments are often repeated, sometimes many times, and for no apparent reason. These microsatellites are highly individualistic, forming a unique DNA signature for each person. Microsatellites have quickly become the most important tool for individual identification, and they have proven especially valuable in the forensic science. For a long time, the general impression about gene has been that they represent specific location of DNA coded to produce specific proteins. Much of research genetics, including in anthropological genetics, is based on this one gene, one protein model. However, the relationship between genes and their physical expression, especially their phenotype, turns out to be more complex than the previously thought. Many traits are polygenic, determined by gene at or two or more loci. However, the genes cannot be identified individually and the physical manifestations are influenced by environmental factors. In human, thousands of traits such as height, skin color, head form, tooth size, and eye shape have multiple genetic components and are strongly influenced by environmental factors. For some of these traits, scientists can determine only the relative proportion of genetic and environmental conditions. A trait heritability, the proportion of its variation that is genetic, can be calculated like heritability is equal to genetic variation divided by genetic variation plus the environmental variation. Heritability estimates are presented as values ranging from 0, where none of the variation is genetic in origin, to 1, where all of the variation is genetic. In traits with heritability estimates greater than 5, most of the variation is genetic. For example, in the United States, the heritabilities for height and weight are estimated to be 0.6 and 0.3 respectively. Tooth size has among the highest level of heritability, about 0.7, and brain size and fingerprints are even higher at 0.9. Physical anthropologists and other evolutionary biologists are very interested in heritability for one simple reason: because only heritable traits respond to natural selection. They are the primary driving force of evolution. Measurement of heritability, however, is complicated by pleiotropy. A single allele can have multiple effects. In fact, most complex traits are polygenic and pleiotropic. This figure shows the polygenic trait and pleiotropic genes. Here you can see that the polygenic trait means many genes contribute to a single effect. Each gene has a distinct biological effect, and phenotropy means a gene has multiple effects. So students, let us summarize what we have learnt in this module. What we have learnt is that much of the differences between humans have a genetic basis. Differences in phenotype caused by differences in genotype. Some of these differences are easily observable, and we know from our own experience that they run in families hair, eye and skin color, stature and some other morphological features. Historically, skin color was one of more frequently explained traits and most systems of racial classification were based on it. Then, taxonomic classification by Linnaeus, cephalic index by Andres Ritz, various physical attributes like skin color, shape of the face, shape of the nose, hair color, hair form and eye color. Since World War II, the emphasis has shifted to the examination of differences in allele frequencies within and between population as well as adaptive significance of phenotype and genotype variation. This shift in focus occurred partly as a result of modern synthesis in biology and because of advances in genetics. Human variation is viewed by physical anthropologists as a result of evolutionary factors such as mutation, genetic drift, gene flow and adaptation to environmental conditions both past and present. Lately, physical anthropologists and other biologists who study modern human variation have largely abandoned the traditional perspective of describing superficial phenotypic characteristics in favor of measuring actual genetic characteristics. Those traits that differ in expression among various populations and between individuals are more important in contemporary studies of human variation. Such characteristics with different phenotypic expression are called polymorphism. Thank you.